We're going to move on with chapter 13, section 3, or point 3. And my goal for you by the end of this podcast will be to be able to list the phases of Meiosis I and describe the events that are characteristic of each phase. And then Objective 8 asks you to describe the process of synapsis during prophase 1. So pay special attention to prophase 1. It's a very unique phase of meiosis. Well, it's important to understand that meiosis reduces the number of chromosome sets from diploid to haploid. There's a cell either in your ovary or your testicle right now that's in girls a long time ago that was going to divide and create eggs, and guys right now that's down in your testicle that's going to divide and create sperm. But the point is that this original cell is diploid, as evidenced up here at the top, and then the final gametes, either the eggs or the sperm, are haploid. So the chromosome number has been reduced. In order to do this, duplication of chromosomes must occur first, meaning, think of a man's testicle and think of a cell that's going to create sperm. In order for that 46 chromosome having cell to create 4 sperm that each have 23 chromosomes, it has to duplicate or create this sister chromatid situation. You can see that here in this second cell on its way to making sperm before it can divide way down into 4 times 23. It needs to duplicate itself up to 92 chromosomes first. Meiosis, you'll see in this whole process, takes place in 2 steps, Meiosis I and Meiosis II. We've already said that 4 daughter cells are made. That's either 4 egg or 4 sperm from one original cell, and that's different than mitosis. And each daughter cell only has half as many chromosomes as the parent cell, and I'll tell you, the chromosomes are not exactly the same as the parent cell. They've been mixed up and stuff in order to give us this variation that I've already mentioned. Here's the picture in your book that you're going to see of 2 chromosomes within a cell, one from mom, one from dad, and then those are going to be duplicated right here in the S phase of interphase, and then those are then going to start to divide out in Meiosis I and Meiosis II. And the key point to understand is that the chromosomes that were in that original cell at the top of this have been divided out, and so now each cell has 23 chromosomes. In the case of humans, each of these gametes has 23 chromosomes rather than 46. So please try to redraw this kind of in the cartoonish way that you just saw, and press pause. Here's what you might have gotten. This is a replication of the cartoon that I showed you earlier. Remember that the chromosomes have to replicate, and that would occur right here in the S phase of interphase, and some sister chromatids would be made in your ovary, girls, or your testicle, guys, before the gametes can be made. And these are genetically identical at that time. Here are the phases of meiosis. It's divided right here down the middle. Meiosis I is on the left, as evidenced by the 1 after all these phase names, and then Meiosis II, or the second half of meiosis, is here on the right. So we're just going to go over Meiosis I right now. Here's the 4 phases of Meiosis I. About 90% of the time of Meiosis I is spent in prophase, and chromosomes are beginning to condense here. Here's prophase over here. This is more metaphase, the second picture. But prophase is here on the left, and you're seeing here that the homologous chromosomes, which have been replicated, they're each now sister chromatids or X's, and they're homologous. Maybe it's the chromosome 11 from my mom and the chromosome 11 from my dad, and I'm preparing to make a sperm. They're coming together, and you see here something called a chiasmata. And a chiasmata means it's an area where the 2 chromosomes are actually touching, and these are exactly the same pretty much. Sorry, not exactly. They're mostly the same. They have the same genes which code for the same characteristics, but those characteristics may be a little different, like brown eyes versus blue eyes. And this is called synapsis when they come together. At the chiasmata, there's this phenomenon called crossing over, and actually pieces of DNA from one homologous pair actually get swapped out with pieces of DNA from another homologous pair. We're going to see a little movie on this in a second, but this happens at synapsis in prophase 1. And what that serves to do is, if you can imagine those kind of mixed up genes being delivered down this whole meiotic process to the sperm or the egg, that those now sperm or egg are going to be a little bit different than the other ones, because there's been this crossing over that has occurred at the chiasmata. Prophase 1 of meiosis is generally the longest phase of meiosis. Why now do you think this may be? We'll talk about this tomorrow. And I wanted to show you this this table because I wanted you to get an idea of how many nucleotides exist in each chromosome. Here are the chromosomes on the left, here are the genes in the middle, and the total base pairs on the right. And wherever that cut is made, like if the homologous chromosome broke off after the 7,011th nucleotide versus the 7,000 and the 12th nucleotide, and then swap that over and crossing over to the other homologous chromosome, that means down the line you're going to get different sperm. And look at how many different places that breaking and off and crossing over can occur. It's pretty amazing, these numbers. Then it gets pretty well. Sorry, there's one more step in meiosis that's kind of interesting, and that happens at metaphase 1. And these now crossed over homologous chromosomes are going to line up at the metaphase plate, but they're going to line up randomly. Maybe the chromosome 11 from my dad is on the bottom, and chromosome 11 from my mom is on the top, and then maybe next to it the chromosome 12 from my dad is up here, and the chromosome 12 from my mom is over here. And the point is as they line up on the metaphase plate, they line up in a random fashion, and it's different every time a gamete is made, either an egg or a sperm. And those then will be pulled in the opposite sides of the cell, the cell that then is going to make a gamete. And then there's going to be more of my dad, say, in this sperm, and more of my mom in this sperm that went on one of those, maybe went on to make my daughter. So she may have more chromosome 12 from my mom or more chromosome 12 from my dad. It's kind of interesting. This is called independent assortment of chromosomes. Anaphase 1's pretty similar to mitosis. They just get pulled to the opposite sides of the developing cells. And telophase 1, the new arrangement is then going to split into 2 cells, and each of these cells actually have a diploid set of chromosomes, not haploid. Cytokinesis then occurs, and then those 2 cells are going to divide again, so that we eventually have 4 gametes at the end of this process. Well, here it is. Make sure that you spend a little time answering these objectives. Thanks. We'll see you tomorrow.